place, and this started back sophomore year 2 years ago. And our goal at the end was to make a prototype that could fit over the eyepiece of a microscope and allow colorblind individuals to see different gram stains, so either pink or purple under the microscope.
Yeah, so this idea kind of started because I myself am colorblind, and Audrey and I got to having a conversation after I had just taken a microbiology final, and I unfortunately was too nervous to ask my professor for help, so I failed a portion of the exam because I was unable to differentiate the colors under a microscope. So I was telling Audrey about my experience, and she was kind of like, there's got to be like some aid that you can get in the classroom or like something to help you. And I was like, I really don't think there is. So we just got into researching it and looking for something that would help, and nothing was out there. So we just saw the problem. We were like, we should do something about this. So that's what inspired our project. And we talked to Dr. Paper, and we got this whole Discovery Day project going with the goal, like Audrey said at the end, to have a prototype.
All right, so let's get into it. So a background on colorblindness. What even is it? So colorblindness is the inability to see color and the entire visible spectrum that most people do. So in our figure, we have the non-colorblind range, which is the full spectrum that majority of people do see. And then we have our range of our 3 types of colorblindness and what they would see normally. So the cones in our eyes, we have red, green, and blue cones. And so colorblindness is when one of these or more of these don't work properly. So it's actually an X-linked inherited trait, and it's carried on our X chromosome, and it's also a recessive trait. So this means that the only way that a woman can be fully colorblind is when she has both recessive alleles inherited from her father and her mother. And the way that a man can become fully colorblind is if his mother is colorblind or carries the recessive allele. So this means that males typically have it. So it's more common in males because they only have one X chromosome. So they only need that one recessive allele to be colorblind.
All right, so this next slide is going to give you an idea of the different types of colorblindness. So our project is focusing on red-green colorblindness, since it is the most common type of colorblindness, and this is also called deuteranomaly over on the left, where greens look more red. So in deuteranomaly, the reds, yellows, greens, and browns can appear similar, and it may be difficult to differentiate similar colors such as blues and purples or pinks and grays. So if you take a look on the top at the bottom row, this one that says range if green cone's defective, that'll give you a little bit of an idea of what a person with deuteranomaly might see. In the middle, we have protanomaly, in which the reds look more green and less vibrant. So if you look at that middle bar, that'll give you an idea of what someone with protanomaly might see. And then over on the right, we have protanopia and deuteranopia. And this is where the person is unable to differentiate red and green at all. So this is the more severe version of the deuteranomaly and protanomaly. But it is also important to note that colorblindness is a spectrum. So each person, even within the individual categories, may experience color completely differently than someone else. So that is very important to keep in mind as we continue with what we found in our research.
All right, so the question that we had was, will incorporating color vision deficiency correcting lenses with microscope lenses allow red-green colorblind individuals to distinguish between gram-positive purple and gram-negative pink bacteria on microscope slides?
So our hypothesis was using color vision deficiency correcting lenses in line with a microscope lens will allow individuals with red-green deuteranomaly colorblindness to distinguish between gram-positive and gram-negative bacteria on microscope slides.
And then our null hypothesis is just that they will not allow individuals to see between this purple and this pink gram-negative and positive slides.
All right, so the next section is our experimental design.
So our design, we set it up with 2 groups. We had our control group and our experimental. In our control group, we had the participants look at all 6 gram stain slides without the color correcting glasses. And then our experimental, we had them look with the glasses. So the conditions that we set up for this were that all of our participants looked at the same 6 gram stain slides to keep it the same throughout every participant.
All right, so here's some examples of the gram stain slides that we used. And as you can see, there are only 2 possible color options, the pink and the purple, and then the third one has both pink and purple, which is why it's a mixed slide. So we blinded our participants to this information and just had them tell us whatever colors that they saw. And then we informed them after the test was finished of the 2 possible options. So we decided to use the gram stain test because it is the first test used in microbiology to begin identifying an organism. And most bacteria can be broadly classified as gram negative or gram positive. So just to give you an idea of how important this test is, an example of a gram positive bacteria would be Streptococcus pneumonia, which I'm sure sounds familiar. And then an example of a gram negative bacteria would be E. coli, which I'm sure you're also familiar with. So there are definitely some safety concerns with mixing up these 2 different types of bacteria. So that is why we are so concerned with this issue. All right, this next section that we're going to talk about is our methods.
So how did we do this experiment? So we had 2 rounds of testing. Our first round, we had them fill out, our participants fill out the IRB informed consent form, which says, yes, we absolutely want to be a participant. And then we had them take a survey that told us how color blindness has impacted them in various environments. So the second thing we had them do was take the N-chroma color blindness test to determine their colorblind type and the severity of their color blindness. And then the third thing we had them do in the first round was look through each 6 microscopes at the gram stain slides without the color correcting glasses, and we recorded their results. In the second round, we had them put the glasses on and then get adjusted to them. Some people took a little longer to get adjusted, which was totally fine. And then we had them retake the N-chroma color blindness test to check if their severity changed at all. And then the last part, we had them look through each 6 microscope slides, and these were with wearing the glasses, and they told us what colors that they observed.
So to organize our data, we would score each participant's responses to the microscope slides as shown on the right over there. So each possible color was worth 1 point. So 1 point for pink, 1 point for purple. And so the mixed slides were worth 2 points for a total of 8 possible points. And then to graph our results, their scores were converted to percentages.
So let's look at some of our results and discuss them. So here's our results table. We had it set up with participants on the far left, types of color blindness that they were, the severity without the glasses, and then the column next to it was percent correct without glasses, and then we did the severity after when they put the glasses on, and then percent correct. So let's look at this in our graph. So here we have it graphed, and we found that there was a 32.14% increase in accuracy when we subtracted our 2 averages. And then we wanted to talk about how all of our participants experienced a decrease in severity. So let's look back at this chart that we made. So we can see in our column that our severity without the glasses were either strong or mild. And then when they put the glasses on and retook the N-chroma test, they were either normal, moderate, or mild. So they decreased in severity, which was a very exciting result. And then a point of interest that we found was that related to severity, even though the severity changed for all of them and decreased, it didn't necessarily mean that they scored better while wearing the glasses. So subjects 2 and 3, they did not have an increase in their score, even though their severity changed. And this relates to some of the challenges that we encountered throughout this experiment. The most important challenge being that when participants put the glasses on and looked at the gram stains, they saw a different color, but they weren't necessarily sure what to call that color because they'd never seen it before. So to help with this in future experiments, we would have a chart labeled with different types of colors so that they could relate and point and say, this is the color that I see in the microscope. So they didn't have to try and explain what they saw, not knowing how to explain it.
All right, so our next set of data that we wanted to share were 2 of our survey questions. So we wanted to share these 2 survey questions and results because they were huge contributing factors to why we started this project and why we think it will be helpful. So the first one was, there's not sufficient aid in classrooms for colorblind students, and this was given on a Likert scale style question. So this one had varied results, with 5 students leaning towards disagree and 4 students leaning towards agree. And then our second question was, I have not felt comfortable asking for help from professors or peers. So this one had 4 participants leaning towards disagree and 5 participants leaning towards agree. So let's talk about that. So as we mentioned at the beginning of our presentation, the goal of this project is to aid colorblind students in the laboratory setting. So the fact that half of our participants believe that there is not sufficient aid is very important and a little bit concerning. So this may make students feel uncomfortable in their learning environment, cause them to receive lower grades, which I myself have experienced, have a difficult time or struggle following the career paths that they're truly passionate about due to their lack of color vision, or even just dampen their excitement for learning due to the lack of aid that is available in the classroom. So as we were researching, we discovered that there really is not a whole lot of aid for colorblind students at all. And in an age of advancing technology, especially in science, and with all of just the color technology that we have now, it is so paramount to learning that students are able to have the aid that they need and access to colors to improve safety and accuracy and be able to follow things that they are passionate about. So the next one was, over half of the participants surveyed have not felt comfortable asking for help. So I myself have experienced this. As I mentioned, I was too nervous to ask for help. And I think a lot of this is driven by the fact that colorblind people know that there's not a lot out there. And this is a problem that needs remedied. And that is the entire goal of our project, is to help colorblind individuals, as many as we possibly can, to save them from these negative experiences. So now we're going to talk about our future hopes with this project.
Yes, so our original goal, like both Mary and I have brought up throughout this talk, was that we want to create a prototype, and we want to create a more advanced prototype. And we also would like to increase our sample size. We realized that with Benedictine being as small as it is and Atchison being as small as it is hard to gather enough participants. So in the future, we would like to test more red-green colorblind individuals, but we would also like to work with other types of colorblindness. So people with deuter anomaly, proch anomaly, and anything else. And then all of these things and more help to enhance inclusion of colorblind individuals in both science and education. So we want to share with you the first mock prototype that we've made, and we will stop screen sharing.
I'll let you do that. We're going to turn off the screen sharing really quick.
That way we can show you guys. Wait. No, let's stop recording. We can figure this out.
We got it.
We'll do that.
All right.
That'll work.
So this is the prototype that we have created thanks to the help of Liam Morrell, the engineering department, and Brookside Optical.
Mm-hmm.
So this is really what our vision was sophomore year. This is what we have been working towards the past couple of years. Yeah. And we'll give you a little demonstration here of exactly how we want this to work. Just this bad boy. So these are essentially just a little sleeve that will go on the microscope eyepiece. So it slides on just like that. And you can even do 2 if there's
compound microscope.
Look at that. So we figured that this was extremely important to have, if possible, in as many labs as there can be. As you can see, I wear glasses already, and wearing colorblind glasses on top of my glasses, I would either have to sacrifice my vision accuracy or my color vision, and both of those are relatively unsafe options. And then you add in the need for safety goggles that are often necessary in laboratories, and it just makes for a bit of a hot mess. So we figured this would be an easy thing for professors or labs professionals to have on hand to ensure that learning